Hi, my name is Rebecca Walker, and this is my eReport for Unit 1 of Cell and Molecular Biology. This eReport has 8 different topics from the first half of our semester. These include chromatin compaction, from protein-free DNA all the way to the mitotic chromosome, as well as the roles of HDACs and HATs in DNA accessibility. The second topic is DNA replication, including everything that happens, every protein involved at the replication fork. DNA mismatch repair is our third topic, with the 2 mechanisms for repairing double-strand breaks. The fourth topic is the assembly and the purpose of the pre-initiation complex. Topic 5 is post-transcriptional mRNA processing. Topic 6 is translation, all of it. Topic 7 is combinatorial control and the mediator. And lastly, topic 8 will cover RNAi. Our first topic, as mentioned, was chromatin compaction. So the first thing that comes up is, what exactly is chromatin? Chromatin can be defined as any packaged DNA. So DNA has to be compacted in order to fit inside the nucleus. An example of this and why it's necessary is just chromosome 22 contains about 1.5 centimeters of DNA when it is linear. Do we think 1.5 centimeters is going to fit inside one single nucleus? No. So it needs to be compacted in order to allow all the information to fit. In order for this to happen, DNA has to wrap around nucleosomes. Nucleosomes are comprised of histones, specifically H2A, H2B, H3, and H4. 2 copies of each histone come together to form an octamer. This allows for a spool shape. 147 base pairs of DNA then wrap around the edge of the spool. Once the DNA leaves one spool, it moves on to the next. In between, there is linkered DNA, and this contains no proteins. Now, you might be wondering, how does DNA bind to the histones? This all has to do with the charge of each. DNA is negative, while histones are positive, which allows for that binding to occur. What I just described was the first stage of compaction. But that isn't compacted enough to be found in a living cell. How about we think of a pearl necklace being wrapped around your finger? The more you twist it, the more it starts to coil. This is an example of the least compacted form of DNA. A 5th histone protein, H1, is what allows us to get to that point. This is known as the linker histone. It sits on the edge of the nucleosome where DNA is exiting. It forces the DNA to go off to one side. This is what allows for more of that twisting or coiling to create the pearl necklace compaction that is desired for living cells. It creates what is known as the 30 nanometer fiber. From there, it is looped and coiled more in order to create a mitotic chromosome, though we're not exactly sure how it gets to that point. Once the chromatin is in place, it doesn't necessarily stay that way forever. Sometimes the DNA will need to be accessed, while other times it may not be relevant. Depending on the need for gene expression, the nucleosomes, histones, and DNA will need to move. Lysines are in the histone tails, and they are what contain that positive charge that allows DNA to bind to the histones. The alteration of lysines can allow for the chromatin to be loosened or condensed. This can be done through HDACs and HATs. HATs, or histone acetyltransferase, are what we see in the red oval of this image. They are responsible for the acetylation of the histones. They add an acetyl group to the lysines, neutralizing the charge. This loosens the nucleosomes and allows the DNA to be more accessible. In the blue oval, we have HDACs, or histone deacetylase, which remove the acetyl group and allow for the positive charge to return, condensing the chromatin yet again. In order for DNA to carry out its functions, it needs to have identical copies. This is where replication is needed. This happens at the origin of replication, but more specifically, the replication bubble. Here, helicase comes in and breaks apart the hydrogen bonds between the base pairs, creating a space in between the 2 strands of DNA. The replication bubble also contains the replication forks. So here's what it kind of looks like at the origin of replication in order to replicate the parental DNA. So we have, as I said, our origin of replication, we have our replication bubble, which again, the helicase came in and split apart those hydrogen bonds between base pairs. And then in our replication bubble, we have the replication fork, which I'll now go into a little bit more detail about. This is a replication fork. It forms on either side of the replication bubble, and the forks move away from each other until they join together. Because of this, DNA replication is known as bidirectional. DNA is antiparallel, and it can only be synthesized from 5' to 3'. As we can see, the top strand is fine. It is able to be made in the 5' to 3' direction, so it is a continuous strand. The bottom strand, however, is struggling a bit more, and it needs to be synthesized discontinuously. The broken fragments are known as Okazaki fragments, as we see in the image. They eventually join together to form a single strand, which we'll talk about in a minute. This is an image of the replication fork in the replication bubble, which is similar to the last image, just a bit more detailed. So we first see helicase breaking those hydrogen bonds in the bases of the parental DNA. Then we need the single-stranded binding protein to latch onto the DNA strands, because once helicase is gone, the strands are going to try to bind together again, but we need them to stay separate for replication to continue. As we can see, there's DNA polymerase III attached to the leading strand. This is what allows for the synthesis of that leading strand, again from its 5' to 3' end. It synthesizes copies of the parent DNA using energy that's broken off from DNTPs. These are the new nucleotides. And it's not pictured here, but there's also a clamp protein which makes sure that DNA polymerase can't float away after adding one single nucleotide. It makes the process a bit quicker rather than having to do one nucleotide, stop, restart the process. It just lets it continue. DNA polymerase needs to bind to an RNA primer in order to add nucleotides, because it needs to add nucleotides to the 3' hydroxyls. It starts at the 5' end, so it needs that RNA primer, which gives the needed 3' end to DNA polymerase to add the nucleotides onto. The leading strand only needs one primer because it's continuous, but because the lagging strand is discontinuous, it needs multiple primers, one at the beginning of each Okazaki fragment. Because of these RNA parts of the lagging strand, it needs 3 more enzymes before it can be complete. The first is primase, which is on the 3' end of the fragment. This essentially chews up the RNA primer on the front of each fragment. The second is a repair polymerase, which puts down new DNA to replace the now chewed up RNA. The last enzyme is DNA ligase, which joins the phosphate sugar backbone to create one DNA strand again. Now, we seem to have 2 new complete strands, which are the exact copies of the parent DNA. Except it's not always like that. Sometimes the wrong nucleotide may be put in. DNA polymerase checks each nucleotide as it goes into the reactor site, just based off of the feel of the DNA. If the nucleotide is smooth, it's assumed that the right nucleotide is in. If it's not smooth, it's assumed that the wrong nucleotide is there. DNA polymerase then shifts the nucleotide to a different part of the enzyme, cutting that phosphodiester bond between the nucleotide and the new DNA strand. It clips the wrong nucleotide off of the strand, and then the process returns to normal. So DNA polymerase should be able to find the wrong nucleotide during replication and replace it. But what happens if this isn't the case? Mismatch repair then comes in to essentially save the day. It takes away about 99% of the errors that DNA polymerase makes. So the mutation gets recognized as a distortion in the DNA, just like with DNA polymerase in replication. The mismatched part of that DNA sequence gets cut out, and then the old strands get used as templates in order to fix the error. So bacteria uses methylation, where they add a methyl group onto the adenine bases of the old repaired strand, which indicates that this strand is good to go. It can be used as a template. The errors have been fixed. This helps to prevent from further errors so that it's not further replicated, translated, etc. Eukaryotes, on the other hand, don't use methylation, but we're not really sure how they tell which ones are good to go and which ones aren't. Another type of issue that can occur is a double strand break. Here is where the DNA molecule is completely cut into 2 pieces, so there's not really a good strand to use as a template. There's 2 different ways to fix it. The first is non-homologous recombination, which is more common, but it's less likable. Homologous recombination is more thorough, but it's just not always possible. So non-homologous recombination takes the two broken ends of the DNA molecule, it blunts them, and it ligates them back together. This fixes the break, but it takes away the nucleotides from the blunting. If it occurs in the middle of the gene can become inactive, which is the loss of function mutation. Homologous recombination, on the other hand, needs a nearby DNA molecule that's almost identical in sequence. So we all have two DNA molecules that are almost the same, which are known as homologous chromosomes, but they're not always near each other in the nucleus, which is why it's not always possible for this strategy to work. Nuclease recognizes the double-strand break, it chews back the overhang, and it creates a longer strand of single-stranded DNA. The strand then probes and samples the nearby molecules for a similar sequence, and if it finds one that's similar, it base pairs with it. Single-stranded DNA molecule displaces one strand and the other molecule to base pair with it, which is known as strand invasion. The branch point is where the two cross. The invading strand is lengthened by repair polymerase, with the other strand as a guide. Once it's synthesized, the branch point migrates, which allows the DNA to be made. Helicase is then used to open up the loop and move the branch point forward. The invading strand is nicked to return to position and then sealed with ligase. This allows us to end with two intact DNA molecules with no lost nucleotides. So it's not always possible, but it is the better option because we're not having that chance of losing nucleotides, whereas non-homologous recombination has a higher chance of loss of function mutations. Our next topic is the pre-initiation complex. So RNA polymerases are needed to start transcription, but they can't do it on their own. This means that general transcription factors, or GTFs, assemble at the promoter with RNA polymerase II. They're responsible for recruiting RNA polymerase to the promoter, positioning it, separating the double-strand DNA, and then launching the polymerase down the gene. Together, these general transcription factors form the pre-initiation complex. I always think of it as DAB-Phe in the order that they join. So first, the TATA binding protein, or TBP, which is a subunit of the transcription factor 2D, binds to the promoter-specific sequence, and this sequence contains many thymines and adenines. These promoters are rich in thymines and adenines because they're weaker and easier to pull apart. So, once the TATA binding protein binds to the promoter, the DNA gets kinked about 90 degrees. This change makes it easier for the general transcription factors to find the promoter region. Earlier I mentioned DAB-Phe, which is the acronym just used to say which order the transcription factors are joining into the promoter region to let RNA polymerase bind and do its job. So as I mentioned, the TATA binding protein comes in first, which is just a subunit of transcription factor 2D. Then TATA binding protein recruits transcription factor 2A and then transcription factor 2B. Once those are in, RNA polymerase is actually able to bind to the promoter, as we see on the right. Then the TATA binding protein recruits transcription factor 2F. Transcription factor 2E then joins and recruits transcription factor 2H. Transcription factor 2H has helicase and ATP, which work to pull DNA apart and make the transcription bubble. The template strand of the transcription bubble engages with RNA polymerase II in the active site, and then RNA synthesis begins. As we know, transcription occurs in the nucleus and has to be exported into the cytoplasm for translation to happen. This basically allows for quality control so that the mRNAs can be checked. They have to be processed, which also includes additions to the end of both sides of the mRNA, which is essentially a stamp of approval. They can only leave the nucleus if they have these additions. The enzymes needed for the additions are on the tail of the RNA polymerase II during transcription. First, the mRNAs get a 5' cap, which is a methylated guanine attached kind of in a weird way, basically upside down. This cap marks the beginning of the mRNA as genuine. mRNAs also get a poly-A tail, which is a bunch of adenine bases added to the 3' end. It requires a number of different enzymes and marks the end of the mRNA as genuine. The two modifications prove that the mRNA is whole and then allows it to exit the nucleus and be translated. They mark the mRNA as a messenger RNA. Proteins that are recruited to the 5' cap and the poly-A tail interact to allow mRNA to circularize. It allows the cell to know that the mRNA is good and fully processed. Most eukaryotic mRNAs also need to be cut into pieces and put back together before proteins can be made because they're interrupted with sequences that are non-coding. This interruption happens in almost all of our genes, and the sequences are called introns, which can range anywhere from 1 to 10,000 nucleotides. The actual coding sequences are exons, though most genes are introns. Introns and exons are transcribed into mRNA because RNA polymerases can't tell the difference between the two. Then, all of the functional exons have to be joined together, while the introns have to be removed. This occurs through splicing. Each intron has a conserved sequence at its ends and in the middle, and the limited sequences are cues for the intron to be removed. A spliceosome removes the introns and ligates the exons together. The freed 5' end is then attached to the A in the middle of the intron, making a lariat, or a loop. There's evidence that introns have been there since the beginning, as they're a magnet for mutations to leave more of the important sequences as healthy, though it's not entirely convincing. We do know one benefit of having introns, which is alternative splicing. There's no rule saying all exons have to be kept, so non-adjacent exons can be spliced together, deleting others and creating new proteins from one single gene. This increases our protein potential and also modulates protein synthesis. Exons encode individual modules of protein functions or domains, and they can be spliced and traded to give proteins a function or take one away. Nothing can leave the nucleus across the membrane without going through the nuclear pore complexes. These only allow mRNA out into the cytoplasm if they have the proteins at the cap, splice junctions, and tail. The RNAs that don't have what they need remain in the nucleus and are destroyed. RNAs that are in the cell for a long time have the proteins made in higher levels. No matter the type, lifetime, quality, or function, all RNAs are degraded by RNA polymerases when their function is no longer necessary. After transcription and the mRNA is processed, the fully synthesized mRNA strand is left in the cytoplasm. So in mRNA, and also DNA, there are codons. These are groups of 3 nucleotides that code for amino acids. These codons can be read in 3 different ways, which are known as reading frames. However, there's only one correct one. This is known as the open reading frame. It starts with AUG, which is the start codon, and it ends with a stop codon that can appear in a few different ways. If the wrong frame is chosen, it can cause the wrong proteins to be coded for, which can often be detrimental. So mRNA meets up with tRNA because the codons of mRNA can't bind directly to amino acids. They need an adapter molecule to bridge them. That's where the tRNA comes in. tRNA has a 3-base anticodon on one end. It recognizes the codon in the mRNA, and it essentially has one that's opposite in basis to that mRNA. The anticodon goes from 3' to 5', whereas the mRNA goes from 5' to 3'. This codon pairs with the complementary codon of mRNA. This allows for the genetic code to be recognized. On the other end of tRNA is a corresponding amino acid. This occurs through charging, where aminoacyl tRNA synthetases charge the tRNA with amino acids. The enzyme then probes the anticodon to see if the sequences match, and then it gets placed on the tRNA. The T of tRNA stands for transfer, so it brings the amino acid to the ribosome. tRNA is self-base pairing, so it's a cloverleaf structure of 4 that eventually fold into an L-shaped structure. There's not a tRNA for each codon, because wobbling can occur. This is where base pairing creates combinations that aren't usually there. For example, a G might pair with an A rather than a C. Ribosomes are needed for translation because they provide coordination. They're a combination of RNA and protein molecules, which form a complex with one responsibility: protein synthesis. Ribosomes are actually 100 times larger than the average protein, and they're very similar in eukaryotes and prokaryotes, in structure and mechanisms. The job of the smaller subunit, which is on the bottom of this picture, is to match the mRNA codons to their tRNA, so they facilitate codon recognition. The larger subunit on the top forms the peptide bond between the amino acids in order to link them into a chain. The 2 subunits come together with an mRNA in the middle. When translation is initiated, it's assured that the mRNA is placed right in the ribosome, near the 5' end of the mRNA. The larger subunit has 3 slots or voids, which are shaped to accommodate a tRNA. One slot is the A site, which is shown as step 4 in the image. It holds the aminoacyl tRNA, which has a single amino acid on them. The P site, which is shown in step 3 of this image, holds the peptidyl tRNA, which carry peptide chains of amino acids. The E site, in step 2 of the image, is the exit site, where the tRNAs leave when their job is done. The site name is named for the type of tRNA that they hold. For translation to begin, the ribosome assembles around the mRNA at its 5' end. In the middle of translation, a tRNA with the correct anticodon as well as the right amino acid enters the A site to bind to the codon through base pairing. We see this in step 1 of the image. Then, the amino acid is held by the tRNA in the A site, forms a peptide bond with the amino acid chain that is hanging off of the tRNA in the P site. This transfers the whole amino acid chain from the tRNA in the P site to the tRNA in the A site. The whole ribosome then shifts forward by 1 codon, while the tRNAs stay base paired with their codons. This is known as translocation. The end result is that every tRNA moves back a site. Only 2 tRNAs can be in a ribosome together, so when a new tRNA enters the A site, the one in the E site leaves. The process is then able to repeat. The mRNA is pulled through the ribosome, and as it moves, codons transit from the A to the P and then to the E sites. The tRNAs go along with them, bringing amino acids in, linking them to a peptide chain, and then leaving the ribosome. This means that the mRNA is decoded 1 codon or amino acid at a time, and the tRNAs are essentially the adapters. This is why tRNAs are considered the translators. Eukaryotic ribosomes can add 2 amino acids to a growing chain per second, while bacterial ribosomes are about 10 times faster. The process continues until a stop codon is reached. During translation, each incoming tRNA enters the ribosome through the A site, but in order for initiation to occur, there has to be something in the P site. To start translation, a specific tRNA called the initiator tRNA first associates with the free small ribosomal subunit. This small subunit then binds to the 5' end of an mRNA, keeping it engaged while the initiator tRNA scans for an AUG start codon, which is also the CAU anticodon. Once the AUG is found, the tRNA base pairs to it, and the larger ribosomal subunit joins the complex and associates. This allows the tRNA to be in the P site. The initiator tRNA is the only tRNA that can enter the ribosome P site directly. By finding AUG, it helps to make sure that the reading frame is correct, so translation can be correct too. The differences between eukaryotes and prokaryotes are in how the cells find and bind to the beginning of mRNAs. In bacterial cells, the small ribosomal subunit carries an RNA that binds directly to the sequence in the mRNA 5' UTR, which is the Shine-Dalgarno sequence. This allows the ribosome to be where it needs to be. In eukaryotes, there are eukaryotic initiation factors, which bind to the 5' cap of the mRNA into the small ribosomal subunit. They recruit everything that's needed for translation, and they make sure everything's correctly positioned. Without the 5' cap, there would be no recruitment site. Translation would not be able to be initiated, and the proteins would be unable to be made. Every protein begins with methionine. Translation initiation is an ideal point of regulation due to its complexity and difficulty. It's a bit easier to terminate translation. It's signaled by stop codons, which release factors bind to. When a stop codon is positioned under the A site, release factors enter the A site and bind to it. No tRNAs recognize the stop codons. The release factors catalyze the release of the polypeptide amino acid chain from the tRNA in the P site. This frees it from the ribosome. The protein can fold into its 2D shape and do what it needs to do. The ribosome tries to translocate, and the smaller unit can't do it, which results in the instability of the ribosome. The 2 subunits dissociate, and that releases the mRNA. Nothing is destroyed. Everything is simply recycled. Enzymes do things because of their shape, and the same goes for RNAs. This occurs through intramolecular base pairing. Only proteins can be enzymes, so these RNAs are called ribozymes. It's 1/3 protein and 2/3 RNA. The RNA component of the ribosome gives it the structure and function to catalyze peptide bond formations. The main function of the proteins is to guide the folding of the RNAs and stabilization. The idea that proteins don't have much to do with ribosomes and their function is because of the RNA world hypothesis. At any one time, a single mRNA is being translated by multiple ribosomes, each at a different point in translation. A new ribosome assembles at the 5' as soon as the preceding ribosome has cleared the runway, so the mRNA is never really clear of ribosomes. This makes it known as a polysome. Eukaryotes and prokaryotes both have polysomes, but only bacterial cells can have transcription and translation going on at the same time in one single DNA molecule. Groups of regulatory proteins work together in order to control a gene's expression. Most of these genes are actually controlled by dozens. The proteins can be activators or repressors. The combination of proteins bind to a combination of sequences. The way that they come together helps to determine if the gene should be turned on or off. This is known as combinatorial control. Each protein essentially has their own vote, almost like a democracy. Some say that the gene should be expressed, while others say that it should be inhibited. But with all of these factors going in, how does gene expression get decided? That's all down to the mediator complex. The mediator is the integrator of the signals for combinatorial control. So activators function to grant RNA polymerase II permission to keep going with transcription and leave the promoter. They don't affect RNA polymerase's recruitment to the promoter, but they do help with transcription initiation. The mediator sits on the end of RNA polymerase II. The relay complex takes the signals, so whether they be positive or negative, activating or repressing, and it integrates them. Once the signals are combined, the mediator then takes a tally and sends the final vote to RNA polymerase II. This is known as a summative signal, and actually any of these signals are enough to tip the scale and change the decision that the mediator sends. It activates RNA polymerase II, which allows transcription to proceed, or it can repress RNA polymerase II, which stops transcription. Now we're on to our final topic, which is RNAi. This is RNA interference. It's used by cells to silence genes, and it's thought to have evolved in order to stop viral infections. So double-stranded RNA molecules need to be formed. Once they're recognized, all double-stranded RNA and single-stranded RNA with the same sequences are destroyed. It's triggered by double-stranded RNA that's fully base paired, which is usually about 21 to 23 base pairs long. The longer double-stranded RNA molecules are cut into the size that they need to be by the enzyme dicer. The smaller fragments are small interfering RNA, or siRNA. They bind to the RNA-induced silencing complex, which is RISC. RISC uses one of two strands from the small interfering RNA to recognize other RNA molecules with complementary sequences. Once it's recognized, RISC then destroys those mRNAs. So RNA interference is actually very potent, but why? It all comes down to amplification. When RISC cuts mRNA down, it leaves two unstable fragments. RNA-dependent RNA polymerase, or RDRP, binds to one of those fragments. It then uses it as a template to generate double-stranded RNA. The double-stranded RNA is used by dicer to generate more small interfering RNAs for RISC to use. RDRP is actually vital to amplification for this reason. So the more small interfering RNAs there are, the more RISC is able to use, and then it's able to destroy those mRNAs altogether, which is why RNA interference is so potent. So this is just an image to give you an idea of what RNA interference looks like. So as we see on the top, we have that double-stranded RNA molecule, and then the dicer is there to break apart that molecule. We're then left with the smaller fragments, or the small interfering RNA, which then bind to RISC, or the RNA-induced silencing complex. Then it's able to use one of the two strands from siRNA to recognize the other RNA molecules with the complementary sequences. So then all of the mRNAs end up getting destroyed. With that concludes my eReport for Unit 1 of Cell and Molecular Biology. Thank you so much for taking the time to watch this, and if you have any questions, please do reach out to me. Thank you.